Everyone, so this is an answer key, if you will, for the Artificial Cell Virtual Lab. Use this as a guide to learn how to provide analysis for experiments like this. So again, we're sort of working in the dark as far as what percentage solution each one of the mystery solutions are. The only thing that you can do to figure this experiment out is to perform experiments and to collect data and analyze the data using your knowledge of what, how diffusion and osmosis work in various types of solutions. So that is, hypertonic versus hypotonic versus isotonic solutions. Okay, so recall these are the experiments that we set up initially. If this doesn't make sense to you, go back to the original video to see, what these solutions are. Basically, you don't know what A is, what B is, or what C is, but it can be either 30%, 10%, or 2% sucrose. So after doing the experiments from the last video, you can calculate the following percent mass changes. So for example, when we place A inside the cell and B outside the cell, you have an initial mass of 10.2, you have a final mass of 8.8, so that represents a 12% mass decrease. So I give you all of the percent mass changes here. Hopefully you've calculated them in a similar manner. When A is inside the cell and C is outside the cell, you get a 12.1% increase. So this increase versus decrease versus staying the same is the data we're going to use to provide our analysis and finally our conclusions as to which of the mystery solutions is 30%, 10%, and 2%. So let's break it down. I'm going to set up all the experiments in this style where we're looking at what's inside, what's outside, and what happens after 1 hour's time. So in this case, when you have A inside and B outside, you lose 12%. Over here, when you have A inside, C outside, you gain 12%. So what does this all mean? Let's break it down experiment by experiment. The first thing I would do in this experiment was to check my controls. Because if your controls don't work out, you really can't trust all the other experiments. So in this case, we did a couple of isotonic controls. That is, A is inside the bag and outside the bag. So whatever these solutions are, let's hypothesize, for example, that A is 2%. If they're both 2%, you shouldn't get a large mass increase. Okay, so this is really just checking to make sure that we did the experiment right and that we didn't mix up our solutions. So in both of these isotonic controls, A inside, A outside, B inside, B outside, you're not getting a very large percent mass gain or loss. That's a good thing. So our controls tell us, let's proceed. Let's see what the other experiments show us. So the next thing that I'm guided to, again, red is a gain, blue is a loss. Experiments in which C is inside the bag both result in percent mass losses. So in both of these instances, when C is inside the bag, it's losing mass. So remind yourself what that means. When solutions are placed in blank solutions, they lose mass. So is it when they're placed in hypertonic solutions, when they're placed in hypotonic solutions, when they're placed in isotonic solutions? When will you lose mass? And the answer to that is when they're placed in hypertonic solutions. So in this case, A, whatever it is hypertonic to C. B, whatever it is hypertonic to C. Okay, so we've established that C is probably going to be the lowest concentrated percentage solution because in both instances when it's placed in A and B, it's losing mass. Now the next thing I'll guide you to is, well, how much mass is it losing? Here you can see this one's losing about 13%. This one's losing about 31%. So again, what that is telling me is that B is probably the highest sucrose solution because you're getting the biggest loss. So that would say, let's hypothesize here, if B is 30% and C is 2%, then it will show the greatest mass loss. And that's exactly what we see here. Now, if C is 2% and A is that intermediate 10%, it'll still lose mass because A is still hypertonic relative to C, but not quite as much. And that's exactly what we're seeing. So you could probably conclude the experiment is done right here, but since we've done all the experiments, let's analyze them and see if we can draw any other meaningful conclusions. The second thing that I'm guided to is, well, there's two experiments here in which both of the experiments result in mass increases. So again, when B is inside the bag in both scenarios here, when it's surrounded by A and when it's surrounded by C, they're both gaining mass. Remind yourself the type of solution that's on the outside resulting in a mass increase inside the bag. Okay, so those types of solutions outside have to be hypotonic. And again, if we hypothesized before that B was 30%, that would make a whole lot of sense, right? So in this case, you have A on the outside, B on the inside. You're getting about 13% increase. Here with B on the inside and C on the outside, you're getting the biggest increase. Okay, so hypothesize again. If C is 2% and B is 30%, you would see the largest percent mass increase. And we see that here, and we see a similar mass increase, but not as large, because again, we're hypothesizing that A is 10%. Last but not least, we have this experiment here, which has A inside. So remember, we're hypothesizing that A is 10%, which is the in-between concentration for this experiment. So if we surround it by what we're hypothesizing is 30% in B, well, then you would see a mass decrease, and you do see it, okay? So here, likewise, if we're surrounding 10% for A with 2% C, you're going to see a percent mass increase because C would be hypotonic relative to A, and that's also what we're seeing. So the answers for this quiz, or for this lab, I should say, B was 30%, A was 10%, and C was 2%. The previous slides show you the evidence that you get from analyzing your experimental results, allowing you to draw the conclusions B equals 30, A equals 10, C equals 2%, okay? I hope this helps. Watch it a couple times if you need to, and feel free to email me with any questions.